Am I audible as well as visible? Let me check. Let me check. Now, let us start today's session. Welcome to Unacademy Future Doctors. I myself, Dr. Deepthi Karya, I'm MD PhD Physiology. I'm associate professor and educating MBBS nursing and physiotherapy students. I have more than 17 years of teaching experience. Now, let me explain something about our subscription. We have two varieties of subscription. One that is plus subscription. Here you can choose the best from the best. In plus subscription, you can access both live and recorded classes. You can study on the device of your choice. You can learn from India's top educators for your medical exams. You can access question bank, which is having more than 25,000 questions. You can compete in the live test as well as quizzes, and within 12 months, we are coming with our printed notes. Okay? Second, that is iconic subscription. Here, your access to the best from two of the best. Which are these two? One that is Unacademy and Prep Ladder with Unacademy. All the features I have already explained. Second one that is Unacademy and Prep Ladder with Prep Ladder. Here the features are you can have clinical and integrated essential, you can have video lectures from the dream team, you can have question bank 3 with active guidance, system tags and more, rapid revision and snapshots, treasures in 2021. So you can subscribe from your mobile either plus subscription or iconic subscription. You can use my code Dipti10 and you can get 10% discount on any of the subscription. Okay. Another thing is longer the duration of subscription, cheaper are the rates. So it is beneficial for you if you are subscribing for long period of time. If you are in first or second MBBS, then you can subscribe for long duration. Okay. Now, one more thing I would like to discuss with you that in this channel we are posting all the videos which are freely accessible. You did not have to pay anything for all these videos, and these videos are for university exam going MBBS students. So if you have anybody in your friends, in your family, or your junior, you can share all these videos with them and they can get the benefit from this channel as well as videos. Okay. Now, so today we'll discuss about humoral immunity. So first of all, we will discuss something about development of immune response. As we have discussed, immunity is divided in two types. One that is innate immunity or natural immunity, and second that is acquired. Okay, this acquired immunity which is developed after birth. That is, we have discussed that is specific for antigen. Means suppose you have antigen, like you have one virus, your body produces antibody against that virus only, not against anything else. Okay? So this is your specific acquired immunity, which is divided in two types: humoral immunity, another name is antibody mediated immunity, and second one is cell mediated immunity. प्लीज कीप इन माइंड बिकॉज समटाइम्स क्वेश्चन ह्यूमरल इम्यूनिटी होता है, आप लोग पूरा सेल मीडिएटेड इम्यूनिटी लिख के आते हो, पूरा क्वेश्चन गलत होता है. ठीक है, आपको पूरा आ रहा है फिर भी ह्यूमरल के बदले सेल मीडिएटेड लिख जाता है. तो दैट इज ह्यूमरल is antibody mediated, or you can say B lymphocytes mediated immunity. Okay, this one. And cell mediated is T lymphocytes mediated immunity. Keep this thing in mind. Okay? Now, humoral and cell mediated immunity. What is the difference? As we have discussed, in humoral immunity, we have B lymphocytes, this one, They produce humoral, and T lymphocytes are responsible for cell-mediated immunity. Here, why is it known as humoral? Because these antibodies, they pass through humor. Humor means our body fluids, like blood. It passes through body fluids. Okay? And it provides defense against bacterial infection. Try to remember this: bacterial and viral infection. Okay? Whereas cell mediated immunity provides defense against viral, bacterial infection plus allergic reaction, allergic reaction, graft rejection. If there is any transplanted tissue, the graft is rejected because of this cell mediated immunity. Okay. Now, so today we will discuss about humoral immunity, one of the very important question. It may be asked as a short note or long question also. Humoral immunity. So if your question is humoral immunity, what you have to write? First of all, this is the immunity which is mediated by antibodies. These antibodies, they pass through humus. Okay. Question may be asked as antibody mediated immunity. So please don't make mistake. If it is antibody mediated or humoral immunity, you have to write down this. Okay. Now, what is the role of humoral immunity? As we have discussed, they provide defense against bacterial infection, mainly this bacterial infection, that is extracellular bacteria. Okay? Viral infection, which infects respiratory tract, intestinal tract. Okay? So in all this, this humoral immunity is important. These antibodies, they participate in immediate type of hypersensitivity reaction 1, 2, and 3 also. Okay? And that is associated with autoimmune diseases also. All of you are knowing autoimmune diseases in which antibodies are produced against our own tissue. So antibodies, that is humoral immunity. Okay. Now, in this humoral immunity, we have two types of responses. One that is primary response and second that is secondary response. Okay. Now, so primary response means whenever any antigen enters in our body, here you can see our body starts producing antibodies. So that is first response. This response requires some time. So this is latent period, this one. Latent period is from 4 days to 4 weeks. Latent period duration is from 4 days to 4 weeks. Okay? And this response is sufficient to limit the antigen. Now these antigens, they are destroyed if these antibodies are produced. Okay? Next is secondary response. You can see here when the same antigen enters in our body, this antibody production is fast. This is secondary response. Okay? And this secondary response, the latent period is less. You can see here as compared to this one. Okay? So more quickly and large amount of antibodies are produced. So this is rapid response because now here we have memory B cells. They produce large amount of antibodies. Okay? Because we have memory of a prior antigenic exposure. Okay? Here important thing is we have to be exposed to the same antigen. Same antigen, then antibody production increases. This is secondary. Okay. Now, stage of humoral immune response. So what happens first? You can see here when antigen enters in our body. First of all, our macrophage, which is phagocytic cell, first it phagocytosis, it causes phagocytosis of this antigen. You can see here it enters inside the macrophage. Okay. After that, it is destroyed and then degraded products, they are presented to our T lymphocytes. Okay. They are present on the surface of the macrophage and which is presented to the T lymphocytes. So this macrophage is known as antigen presenting cell. So processing of antigen, and this antigen is presented for production of antibodies. Okay. Now, next is recognition of antigen by lymphocytes. Now, how this lymphocytes they recognize the antigen? Okay. T and B lymphocytes, they have receptors. T lymphocytes, they have T cell receptors present on the surface, whereas B lymphocyte also have particular immunoglobulin, as we have discussed, IgD and IgM. They recognize the antigen, and accordingly there is multiplication of T and B. Okay? So you can see here, after it binds with the lymphocyte, now this lymphocyte gets activated. You can see here, this diagram is very important. You can draw this diagram also. Now I'll explain. First of all, you can see this antigen enters in our body. Okay? Now this antigen is phagocytosed by macrophage here. Okay? And after that, what happens? This macrophage has MHC1 and MHC2 receptors, to which this antigen binds. The destruction products of this antigen binds. It is as easy as, suppose अगर आपके घर में, आपके फ्लैट में कोई चोर आ रहा है, तो सबसे पहले क्या करेगा? वो आपका गार्ड है, वो उसको, चोर को पकड़ेगा और उसको पुलिस के पास ले जाएगा. सेम थिंग, मैक्रोफेजेस आर आवर गार्ड्स. They just catch the antigen, catch the thief, and present it to the police. Here, police is our lymphocytes, okay? T and B lymphocytes. So these are known as antigen presenting cells. Now, so this are presented to T lymphocytes. Okay. Now T lymphocytes are converted into cytotoxic, proliferated and multiply, and they form cytotoxic, और दूसरा नाम उसका है killer T cell. Kill करते हैं. ठीक है. And the second type of T lymphocytes are regulatory or suppressor T cell. Suppress करेंगे activity. Okay. So here this way they will act. Second thing, this macrophages, they also produce interleukin 1. ये जो interleukin है, that stimulates production of B lymphocytes. B lymphocytes क्या करेंगे? They produce plasma cells. Plasma cell produces antibodies, and memory cells are also produced. Okay, so this way our body react with the antigen. Okay, so here you can see the same thing which we have discussed. First, another important thing, this macrophages, you can see here macrophage. Macrophage के ऊपर जो MHC2 particles है, yesterday I told you in the class, MHC2 particles, they present antigen to helper T cell. Here these are helper. मैंने बताया था 2 में ऐसे 2 होता है तो helper लिखना है. ठीक है. And this helper T cells, you can see here, these are helper T cells. They produce interleukin 2. This interleukin 2 stimulates B lymphocytes. So you can say that T lymphocytes are required for activation of B lymphocyte, and this is known as T-B cooperation. If B lymphocytes are absent, T lymphocyte cannot. If T lymphocytes are absent, B lymphocyte cannot be produced. Am I clear? Now, next is activation of B lymphocytes. As I told you, same thing. B lymphocytes are proliferation. Proliferation means multiplication, and transform into two types of cells, plasma cells and memory cells. Now, plasma cells, you can see here, they become large. They have large amount of granular endoplasmic reticulum, and they produce antibodies. And small amount of memory cells are also produced. Memory cells, they remain in the lymphoid tissue. एक बार बन गए फिर वहीं पर रहेंगे for lifetime, a long period of time. So second time when same antigen comes, this memory cells, they produce antibodies. Okay, so इसीलिए secondary response होता है वो बड़ा होता है. Secondary response क्यों? Memory B cells होते हैं इसलिए. Okay. Now, एंटीबॉडी प्रोडक्शन, जैसे मैंने बताया प्लाज्मा सेल्स, हियर यू कैन सी प्लाज्मा सेल्स है ना, इसमें से एंटीबॉडी बनेंगे, दिस आर प्लाज्मा सेल, दिस आर एंटीबॉडीज। एक प्लाज्मा सेल अराउंड 2000 एंटीबॉडी मॉलिक्यूल्स बनाता है पर सेकेंड। ठीक है, प्लाज्मा सेल की कैपेसिटी। अनदर इम्पॉर्टेंट थिंग, ये जो प्लाज्मा सेल है जिससे एंटीबॉडी बन रहा है वो स्पेसिफिक होते हैं। स्पेसिफिक मीन्स सपोज कोविड वायरस है तो जो एंटीबॉडी बनेंगे वो कोविड के अगेंस्ट ही होंगे, नॉट अगेंस्ट सम अदर वायरस। सो दे आर स्पेसिफिक। सॉरी। नाउ प्राइमरी एंड सेकेंडरी रिस्पॉन्स, आई टोल्ड यू प्राइमरी रिस्पॉन्स में एंटीबॉडी कौन से होंगे, IgM टाइप, और सेकेंडरी में IgG टाइप। प्राइमरी रिस्पॉन्स में एंटीबॉडी लेवल इज हाई, सॉरी इट इज लो, सेकेंडरी रिस्पॉन्स में इट इज हाई। हाँ, देयर इज वन मोर थिंग, ये शॉर्ट नोट भी पूछते हैं और शॉर्ट क्वेश्चन, क्लोनल सिलेक्शन थियरी। ये थियरी बर्नेट ने दी थी 1957 में। तो ये थियरी क्या बताती है कि जो स्टेम सेल्स है जिसमें से हमारे लिम्फोसाइट्स बनते हैं, ये डिफ्रेंशिएट करते हैं मिलियंस ऑफ T एंड B लिम्फोसाइट्स में, और हर T एंड B लिम्फोसाइट स्पेसिफिक टाइप के एंटीबॉडीज या T लिम्फोसाइट्स बनाता है। ठीक है, तो number of B lymphocytes capable of reacting with different antigens they are produced. Okay? These are identical, immunologically and genetically identical. इसका मीनिंग क्या है, ये एक बी लिम्फोसाइट में से जितने भी बी लिम्फोसाइट्स बनेंगे सब इसके जैसे ही होंगे। सो व्हाट इज क्लोन? क्लोन मीन्स, यहाँ पे लिख सकते हो उसका डेफिनेशन अगर पूछे तो, पॉपुलेशन ऑफ सेल descended by asexual reproduction. asexual means reproduction डिफरेंट टाइप का होगा, एक में से दो में से चार ऐसे बनेंगे, फ्रॉम सिंगल सेल, एक ही सेल में से जब बनते हैं मल्टीपल सेल्स विदाउट सेक्सुअल रिप्रोडक्शन तो उसे कहते हैं हम क्लोन। जैसे कि एक टी लिम्फोसाइट्स है उसमें से मल्टीपल टी लिम्फोसाइट्स बनेंगे। अनदर इम्पॉर्टेंट थिंग, अगर ये हमारे एंटीबॉडीज हमारे बॉडी के अगेंस्ट, प्रोटीन के अगेंस्ट बनेंगे सेल्फ, तो दे आर डिस्ट्रॉयड, वो डिस्ट्रॉय हो जाए। So that is to prevent autoimmunity. Okay? Now, what is attack और इफेक्टर फेज? अभी ये जो एंटीबॉडी है वो कैसे काम करेगी? इनएक्टिवेशन ऑफ एंटीजन, sorry. There are two ways of inactivation. One is direct attack. ये जो एंटीबॉडी है वो एंटीजन के ऊपर डायरेक्टली अटैक करते हैं, एंटीबॉडी एंटीजन के ऊपर, दिस वन। एंड सेकेंड थ्रू कॉम्प्लीमेंट सिस्टम, ये बहुत ही कॉम्प्लिकेटेड है, प्लीज ध्यान रखना है। ठीक है, वी विल डिस्कस वन बाय वन। पहला है डायरेक्ट अटैक। डायरेक्ट अटैक में चार मैकेनिज्म है, चार रिएक्शन होते हैं। पहला रिएक्शन होता है एग्लूटिनेशन, एंटीजन और एंटीबॉडी का जो कॉम्प्लेक्स बनता है वो एंटीजन के सरफेस, सेल के सरफेस के ऊपर है, यहाँ पे, जैसे हमने ABO ब्लड ग्रुप में देखा, और दे बिकम एग्लूटिनेटेड, उसका एग्लूटिनेशन होता है और फेगोसाइटोसिस हो जाता है, डिस्ट्रक्शन हो जाता है, पूरा सेल डिस्ट्रॉय हो जाएगा। तो पहला मैकेनिज्म एग्लूटिनेशन। दूसरा मैकेनिज्म है प्रेसिपिटेशन। व्हाट इज प्रेसिपिटेशन? दिस एंटीजन एंटीबॉडी जो कॉम्प्लेक्स बन रहा है, एंटीजन एंड एंटीबॉडी कॉम्प्लेक्स, इट फॉर्म्स इनसॉल्युबल प्रोडक्ट। तो जो हमारा एंटीजन है जो कि सॉल्युबल है उसको इनसॉल्युबल कर देगा, so it can be easily removed. थर्ड मैकेनिज्म न्यूट्रलाइजेशन। न्यूट्रल मीन्स उसको नलीफाई करेगा। तो जो भी एंटीजन है उसकी टॉक्सिक इफेक्ट्स है, ये टॉक्सिक इफेक्ट्स आर नलीफाइड। एंड फोर्थ वन साइटोलाइसिस, डिस्ट्रक्शन ऑफ द सेल। यहाँ पे आप देख सकते हैं, हियर यू कैन सी एंटीबॉडीज They bind with the cell and they cause destruction of the cell lysis. Okay? Now, second is through complement system. Now, what is this complement? Complement क्या होता है? This is a system of 11 proteins जो enzymes की तरह भी काम करते हैं. Names are C1 to C9, B and D. They are present in our tissue fluid. They are in inactive form. They act by this complement system action by two ways, classical pathway, alternate pathway. Now, classical pathway में क्या होता है? सबसे पहले एंटीजन बाइंड्स विथ एंटीबॉडी. हियर ये जो अपना एंटीबॉडी का कौन सा रीजन था? वेराइबल रीजन और FAB एंटीजन बाइंडिंग रीजन, इसके साथ बाइंड होगा. ठीक है. आफ्टर इट बाइंड्स ये C1 प्रोटीन को एक्टिवेट करेगा, कॉम्प्लीमेंट के C1 to C9 प्रोटीन. ठीक है. C1 फिर C2 एंड C4 को एक्टिवेट करेगा, देन इट एक्टिवेट C3. विल डिस्कस. You can see here. This is the diagram which is more informative. You can see. सबसे पहले first one, antigen antibody complex. You can draw this diagram in your exams. From any of the other diagram, this is the most authentic diagram from Guyton. I have taken this diagram from Guyton. So you can draw this diagram, draw this one in exam. Okay, ठीक है. So first of all, here you can see antigen antibody complex. It activates C1. यहाँ पे you can see here this is a line. So this is active. Now this active C1 activates C4 plus C2. So you can see here this forms C42 activated plus C4A. C4A, then C42 activates C3 and this C3 is converted into C3B and C3A. C3B activates C5. C5 is converted into C5B and C5A. This dash is active. C5B activates C6 plus C7 and it forms C5B67. C5B67 activates C8 plus C9 and it forms C5B6789. Important functions. This C4A, C3A and C5A. A for A, you can say activation. Their function is activation of mast cells and basophils. This is the function of C3A, C4A, C5A. Okay. Function of C3B, B for B, opsonization of bacteria. Opsonization means easy meaning, make it tasty. Easy example. Suppose you cannot eat raw things. आपको आटा दे दे तो खा नहीं सकेंगे. हम क्या करते हैं? उससे रोटी बनाते हैं. सेम थिंग. जो बैक्टीरिया है उसको कवर करेगा और उसको make it ready for phagocytosis. That is opsonization. Okay? So C3B causes opsonization of bacteria. And C5A causes chemotaxis, chemical attraction of WBC. And last one that is formed, that is C5B67, it causes lysis of the cell. Okay? So these are the actions through complement system. Very, very important diagram. You can draw this diagram along with the video or you can take screenshot, but this diagram must be there in your answer. Okay? Now, so here this already we have discussed. Opsonization of bacteria is the function of C3B. We have discussed. Lysis is the function of C5B6789. Okay? Then chemotaxis, chemical attraction of WBC is the function of C5A. Okay? So these are all the functions through your complement activation. So this is the same thing we have discussed. Last complex that is called cytolysis. Okay? Okay. Now, next is alternate pathway. Here, what is alternate pathway? Here what happens, there is some protein which is present in the circulation. The name of this protein is properdin. Okay? Name of this protein is properdin. You can see here, this is a protein, properdin. Okay? This protein present in our circulation and it binds with polysaccharide. This polysaccharide is present on the cell wall of the organism. Okay? And when this protein binds with bacteria or any other organism cell wall, it directly activates your C3. Okay? So C3 is activated and then other reactions will be there. You can see here. This is microorganism. When it binds with B and D proteins and properdin, then it directly activates this C3. Then all the other steps will be followed. Okay, so C3b, C3 is converted to C3b, C3a. Then C3b activates C5, C5b activates C6 plus C7, C5b67 activates C8 and C9, and it forms a complex which causes lysis of the cell. Okay. Now, clinical aspect here. C-reactive protein. What is this C-reactive protein? Sometimes may be asked as a short question. Now, what is this C-reactive proteins? कोविड में बहुत बार देखा है ना, टेस्ट करवाते थे CRP. What is this CRP? So this C-reactive protein is produced from our liver when antigen enters in our body. Okay? So the C-reactive protein, it acts as, what is the action of C-reactive protein? It covers antigen and then it activates complement path, and this complement causes destruction of the antigen cytolysis. So this is the mechanism of C-reactive protein. Then next is interferons. What is this interferon? When any cell gets infected by virus, you can see here, this virus enters inside the cell. Okay, and now our cell produces this interferons. These are interferons. Now this interferon binds with the other cell, and these interferons, they give signals to the nucleus. Now nucleus produces antiviral proteins. Okay, so they protect our body from the virus infection. So when virus enters in the other cell, these particles, they are destroyed easily when we have interferons. Okay, so certain antiviral proteins are produced by this interferon and they prevent replication, multiplication of virus. Okay, so this is all about humoral immunity. Questions asked from today's topic, they are, one is humoral immunity, okay, or other way, that is antibody mediated immunity. दोनों में से कोई भी आएगा, write down the same whole today's topic. Antibody mediated immunity and humoral immunity may be the question. Okay. Short notes may be asked here, that is, this is role of leukocytes immunity from today's topic. This is the question, humoral immunity. Okay. Now, thank you. Thanks for listening. If you like my video, you can press the like button. You can share with your friends, with your juniors, and you can subscribe our channel. Okay. If you are post MBBS exam going students, please watch all these videos. These videos are very important for your exams. They help you to write down answers in your exams.